Hello class, this is our final installment of cellular physiology, and we're going to try to put a bunch of stuff together in this particular section. So I want you to think about epithelial cells. Epithelial cells have a lot of transporters, and they have to be hooked together into a layer, and certain proteins are going to be on one side of the cell and other proteins are going to be the other side of the cell, and we're going to put some of that stuff together. We're also going to talk about fluid compartments that are lined by epithelial cells and compare them. What's going on with the plasma versus the interstitial fluid versus intracellularly. We'll come right back to homeostasis, and finally we want to talk about something called the Donnan effect. All right, looky here. Here's some epithelial cells. You see one and part of one and part of one. Now notice, they sit on a basement membrane. So there's an acellular layer that gives them some structure. And what I really want you to see is, where the heck is my A? This structure along here, I want you to notice how three-dimensional it is. This is what's called a tight junction. Tight junctions hook epithelial cells together so that they form a sheet. And the analogy that I think of is, imagine a six-pack of Diet Coke, or the beverage of your choice. The top of the can, where the pop top is, that's what's called the luminal side of the cell. That's the luminal side of the cell. That's pointing to the empty space, or the lumen. Some people call that the apical cell, the apical side of the cell. So that's where the microvilli are. That's the top of the cell, essentially. And then, getting back to our Diet Coke cans, you buy it in a six-pack, there's that six-ring plastic thing. That's like a tight junction. So many pictures show you this part and this part, and it's so two-dimensional. I need you to understand that the tight junction goes all the way around the cell and hooks all the cells together in a sheet of cells. Then we have what's called a basolateral side of the cell, so the sides of the cell and the bottom of the cell. This is pointing in towards the body. So, for example, the apical side points in towards the blood vessels, and the basolateral side would be, on the basement membrane in the wall of a blood vessel. The apical side is pointing towards the lumen of the GI tract, and the basolateral side is sitting on the submucosa. The apical side is pointing out to the kidney tubules, and the basolateral side is sitting on the basement membrane close to other tissues. The apical side is pointing out towards the lumen of an alveolus, whatever it is. So we see lots of these kinds of arrangements. I just need you to see that how three-dimensional the whole thing is. So there are tight junctions that hook cells together, and we see a couple of ways things can get through cells. They can go, whoops, I lost my pointer there. Here we go. So something can go through the cell. They can cross the apical side and then cross the basolateral side. That's transcellular transport. But there are some tight junctions that aren't that tight. And so things can kind of squeeze between the cells. That's called paracellular transport. So if you think about the epithelial cells in the duodenum, the upper part of the GI tract, the tight junctions aren't as tight as they are down towards the end of the GI tract. Not as tight as they are in the colon. Tight junctions in the bladder, really, really tight. Tight junctions in the stomach, really, really tight. Tight junctions in the proximal tubule, not so tight. Tight junctions in the distal tubule, really, really tight. So their characteristics can change a little bit. Another thing that can connect cells together are things called gap junctions. Notice that this cytoplasm is in actual communication with the cytoplasm of the next cell. Gap junctions are so important in electrical tissues like the heart and smooth muscle. In fact, in the heart we have these structures called intercalated discs. They physically and electrically connect the cells together so that they're in perfect communication. So that's where we're going. We start out with a single cell, but then cells have to be put together in a tissue, and lots of cells have to be functioning together. So if we compare a couple of different kinds of tissues, epithelial cells are closely packed. They don't have a lot of extracellular matrix. They're sitting on top of a basement membrane. So there's sheets of cells held tightly together with tight junctions. There's an apical side of the cell, or a luminal side of the cell, and a basolateral side of the cell. And the blood supply tends to be underneath, and oxygen diffuses up to the cells. Connective tissue, the cells are really spread far apart, and there's lots of extracellular matrix. So in your tendons, in your ligaments, connective tissue in the layers of the gut and whatnot. Bone is a kind of connective tissue. Cartilage is a kind of connective tissue. And they mostly have nerve and blood supplies. Cartilage relies on diffusion. Great diversity of types. So here's my little paint program picture of an epithelial membrane. Whoops! I want you to notice, here's the basolateral side. This is where the sodium-potassium ATPases are. On the luminal side is where you're going to find some other transporters. Here's a secondary active transporter. Sodium comes in, hydrogen goes out. So sodium is being pumped out on the basolateral side to generate a sodium gradient. That sodium gradient allows us to pump out hydrogen. The hydrogen is actually made within the cell. I'm not ready to go there yet. And then the hydrogen can be pumped out. Sometimes there are active transporters. Now it happened again. Sometimes there are active transporters for hydrogen, or hydrogen-potassium. Hydrogen's going one way, potassium's going the other way. Here's an interesting example. So here's our SLGT, our sodium glucose transporter. So on the basolateral side of the cell, we have a sodium-potassium ATPase making a sodium gradient. Then the SLGT uses the sodium gradient to make a glucose gradient. And then the glucose leaves the cell by a facilitated diffusion transporter, or a GLUT transporter. Now this particular arrangement is found in two places in the body, in the gut and in the proximal tubule. So the purpose is glucose is being taken all the way across the cell. I showed you an example of a facilitated diffusion transporter where glucose came in the cell and was immediately phosphorylated. Most cells just have the GLUT transporter because they're just trying to get glucose in the cell so they can use it. But here we're trying to get glucose across the whole cell layer, because either the person's eaten a donut and they have glucose they need to absorb, or the person has filtered some glucose and they don't want to pee the glucose out. They want to reabsorb it and return it to the vasculature. So here's a wonderful example of how we have primary active transport, secondary active transport, and then facilitated diffusion all in the same cell. And looky here, we have our tight junctions holding the cells together into a layer. This isn't a real cell layer. I just took 3 epithelial cells and crunched them together in the picture so that I could show you how these transporters can work together. Now, some of our body fluids are inside the cell, some are outside the cell, and how does that all work? Well, we're around 60% water. Sometimes people say, oh, we're 90% water. No, that's complete horse hockey. We're about 60%. If we have more body fat, I'm not mentioning any names, but if we have more body fat, we have a little less water percentage. Maybe we're more like 50%. And water is a very important part of homeostasis, and we have to balance the water we take in. We can drink water, we can produce water through oxidative phosphorylation, and the water that we output. So the water that we sweat, the water that we exhale, the water that we produce in the feces and the urine. We got to have a balancing act. So about of our body water, 3 liters are in the blood vessels. That's the plasma compartment. About 11 liters are outside the cells in the interstitium. But most of our water is actually inside cells. However, what's homeostasis regulating? It's regulating the extracellular fluid, either the interstitial fluid or the plasma. And the idea about homeostasis is if we regulate the outside of the cell, then the inside of the cell is going to be regulated indirectly. So I'm going to show you a table. I want you to look at it and see what you notice. I have adapted and stolen this table from a textbook called Guyton and Hall. So notice, volume in liters, 3, 11, and 28, 4%, 20%, 40%. Protein, look where your protein is, a lot in the plasma, but most of your protein's inside your cells. Where are the high energy compounds? Inside the cell. Where's your calcium? Outside the cell. Where's your sodium? Outside the cell. Where's your potassium? Inside the cell. Where's your magnesium? Inside the cell. Where's your bicarbon chloride, your anions? They're outside the cell. Where's your phosphate buffer? Inside the cell. And so we have, when we add up all these particles together, we get osmotic pressure. Notice that the osmotic pressure is a bit higher in the plasma compartment. And that's because you have a lot more protein in the plasma than you do in the interstitium. So that's the basis of something we call oncotic pressure. And we will talk about hydrostatic pressure and oncotic pressure when we get to the vascular system and the capillaries. Now, that plasma contains a protein called albumin. And that albumin is a protein that's exerting oncotic pressure. But something else is going on. Now, oncotic pressure is a pressure that pulls into the vessels. So albumin is like a big sponge. It holds fluid in your capillaries. When people don't have enough albumin, they have a hard time maintaining blood pressure. They have a hard time maintaining fluid. So you see these little children who are malnourished, and they have the kwashiorkor. They have all that fluid in their belly. That fluid is supposed to be in their blood vessels, but it's collecting in their belly because they don't have enough protein in their blood vessels. The thing is, albumin is even more powerful than you originally thought. Maybe you didn't know how powerful albumin is, but albumin is powerful because it's charged. So for every albumin, it's got maybe 18 or 19 charges on it. So for every albumin, there are another 18 or 19 sodium. So instead of one osmotic particle, there are 20. That intensifies the oncotic pressure effect of albumin, and that is called the Donnan effect. So when you lose one albumin, you also lose 19 sodium ions. And that's a very important consideration. And our nurse anesthesia students can attest to the idea of how do you manage blood pressure? How do you manage people that don't have enough fluid in their blood vessels? Do you just give them, saline? Do you give them fluids that have kind of fake albumin in it? Do you give them albumin fluids? And the research has been kind of interesting about this. One of the things we're doing in this course is I want you to have an appreciation how knowing about basic science can reveal why things are done in the clinical arena. And I truly hope you're able to make some of those connections in this course. So let's just wrap up sort of a big summary when we think about this table. The extracellular fluid is high in sodium, high in chloride and bicarbonate. The intracellular fluid is high in volume, high in potassium, high in phosphates. that is something you should have committed to memory. Now, you need to remember that sodium is 140 millimolar in the plasma compartment. No, I'm not asking you to memorize that, but I hope that as you go through this course, you start getting a number sense. If somebody said, oh, my sodium's 100, you should say, what? Whoa, that's low for a sodium. Why aren't you having a seizure? I mean, you know, it's that... I'm kind of making a joke here, but I want you to appreciate is that it's a very powerful thing to get a sense of what a value should be and recognizing when a value is not what it should be. And we're going to be working on those kinds of things through the whole semester and next semester too. So I believe I've come to the end of this presentation. All right, so 5 presentations. We're so excited about learning about cells. I hope you are anyway. And I'm going to end it here. Remember, look at your learning objectives, identify what your questions are so we can get the most out of class. Take care, everybody. I'll see you later.